So thank you to Göknur for a last-minute change to the program. So she's going to present the final talk of Biopsy Asia, and it's a talk on back to CRISPR. So back to what we were talking about in the very first session of the conference. So over to you, Göknur.
Thanks so much for organizing that shift, Peter. It was very helpful. Sorry for delaying my talk. So today I'll be talking about a CRISPR Web Analyser tool that we recently developed. And it is to analyze pooled CRISPR screens using Agile Pipeline. First part of my talk, which is just a couple of slides, is describing the CRISPR systems. And the second part is showing basically what the workflow is to design and execute these systems and analyze the systems. And the third part is basically the main part of my talk, is showing you an example data analysis using CRISPR Web Analyser. So the first part is what the CRISPR is. Possibly you've heard of it, but just briefly, it's a bacterial defense mechanism. It's like a genome editing described in bacteria first, and it has two phases. One of them is immunization, when a virus attacks a bacteria. Cas enzyme here just catches this piece of DNA, the DNA of the viral DNA, and writes it inside its own bacteria's own genome to use later, if the same virus attacks again, to use this piece of DNA sequence to destroy the virus. That's basically how CRISPR works, very simply. And CRISPR systems has plenty of modalities at the moment, but most developed ones and most commonly used ones are knockout and knock-in screens. So what they do is to edit the DNA, Cas nucleases form double-stranded DNA breaks, and these breaks are fixed by DNA's own DNA repair mechanisms. And for these mechanisms, non-homologous enjoining or homology-directed repair systems are used. When the first one is used, we call them like CRISPR knockouts. And the other one is homology-directed repair, where we use a template donor, then they are the CRISPR knock-in screens. We will be focusing on CRISPR knockout today. So what are the workflows? There are two workflows. One of them is designing and executing the CRISPR screens. And to design your screen, first of all, you have to decide what is suitable for which guide RNA library is suitable for your experiment. You can get that from a database called EdGene, or you can be collaborating with people and they can send you the viruses. And secondly, you want to check if the quality of these guide RNAs are okay. To do that, you just sequence them and you want to see a normal distribution, so they're all randomly expressed. And the last step is designing your experiments to maintain single guide RNA representation. You will have a certain amount of viruses and you want to have certain amount of cells, and you want just one guide, which means like virus targeting each cell. So you design your experiment to achieve that, and you apply your drug treatment or whatever your experiment is, and then you sequence your data. And it comes to us, computational biologists. And this is the second workflow where we use to analyze this data. First, we get the NGS data as a FASTQ file, and we do align them, but we don't need to use any counting algorithm. You can just count them as they are. And they're not complicated. They're just like 20 base pair amplicons. You're just counting how many amplicons there. And the second part is preprocessing the data, and then we identify the hit genes. So let me walk you through this example data sets by using our tool. The data is something we analyzed in 2020 and published with James Whittle and Jeffrey Lindman from WEHI. They conducted a genome-wide CRISPR knockout screen in different cell lines. Their experiment was to understand the mechanisms impacting the triple treatment applied to one subtype of a breast cancer called ER-positive. So they took those ER-positive cell lines and they applied 3 different drugs. So the vehicles are just the initial guides, Cas-expressing cells, and fulvestrant is one of the treatments. ABT 199 is the venetoclax, it's the BCL2 inhibitor. This is our target treatment here, and palbociclib is another treatment. So what they've done is they combined these treatments with each other, and they observed the doublet treatments and triple treatments. They wanted to see the impact of those treatments. So what they observed is they have seen they couldn't reduce the cell colonies when they applied doublet therapies, but when they added the venetoclax into this doublet therapies, they show quite a big reduction in the cell colonies. And even though they did stop giving the treatments, the cell colonies appeared back on the cells treated by doublet therapy, but no or very minimal growth for triplet therapy. So they wanted to understand what are the main mechanisms behind that triple therapy. So we conducted, they done experiments, we got the data, and we did, here is the library size for this data. So as you can see, we haven't had any much cells left in the triplet therapy at the purple Wyland plot, but we can still see the expression level of the remaining guides are really high. That's the case for CRISPR screens in general. So you have lots of zeros or very low counts, and on the other hand, you have very high counts as well. So you have bimodality in the data. So what we do first is we do align the counts, and when you align the counts, you can use R-subread or bowtie or any aligner, but in some guide RNA libraries, you will have different size, different lengths of single guide RNAs, like not all the guide RNA are going to be the same size. Then it is best to use an alignment-free quantifier if your guide RNAs are differing in size. So let me start talking about CRISPR Web Analyser. This is our tool. We have 4 tabs. One of them is providing you what type of data you have to provide, and it shows you all the descriptions of the samples and the gene data frames. How do they need to look like? And in the second part, you can check the quality of your data. After you're uploading your data, you'll have these options. So you can visualize a data table showing you all the row counts, or you can check the single guide RNA distribution. And you can also check if your library, with the cumulative distribution, you can check if your library is randomly distributed, normal enough to use. This is very preliminary. This is used for very preliminary analysis. And you can see the distribution for each gene as well. And you can see the heat map of 0 counts. That's something we found very useful, and it is invented by Luke Gandolfo from WEHI Bioinformatics. This is different than a heat map. You see, this is basically a binary heat map. Just the yellows are representing the 0 counts, and blues are representing the non-zero counts. We just want to see the sparsity of the data. So when we look at the first column, which is the reference column, reference samples, we do see like quite a random distribution as we expected. with some sort of non-zero counts. But when we look at the treated samples, we can see a lot of zeros in some of the conditions. So heat map 0 is very useful, and we use this for our purpose of filtering. So next step is filtering. In an ordinary RNA-seq experiment, when we filter the row counts, we remove this spike and spike of the frequency of the low counts, and then we obtain a unimodal distribution, but that's not the case for CRISPR. So CRISPR Web Analyser provides you with this filtering options. So no filter, you can weave the data as no filter, and you can weave the density plots for only removing the zero counts, or you can use a filter by expression function from HR, and you can see the distribution after this filtering. So you can see filter by expression filtered down to 17,000 guides, while the initial number of guides is 118. So it trims quite a lot of guide, but it still has this spike up here. So it's still keeping us of lowly expressed guides. So we wanted to propose a new filtering approach. Then we did propose this new filter approach. So we kept more guides in the analysis, and most of the guides we removed were the guides with low expression. So how does this new filtering works is simply, in the previous one, you will see this dialog box here. And in here, we are just defining the treated samples here. The reason we select those is because this is how this filtering algorithm works. You define what your treated groups are, and out of those, you select the group with the highest number of zeros, and then you mark this as the kept group and mark rest of the samples as unkept group, and you define a threshold, two thresholds for both conditions, and then that's how we simply defined our new filtering approach. A bit more sophistication there, but I can't go into details now. And then you obtain a bit better filtered data. So next step is normalization. In the data pre-processing step, pre-processing step, there's all the normalization methods provided in EDGAR is provided here. And what we also observe is we don't see much difference by applying any normalization methods to CRISPR data, and that seemed to be the case for many CRISPR data sets that I analyzed. So my conclusion is a new normalization approach has to be developed for CRISPR data. And but you can visualize them using the Web Analyser. And another thing I want to show you is after you also apply the differential expression analysis, what we are seeing in these figures are the p-value histograms. So at the columns, we have all different type of normalization methods, and at the rows, we have the different filtering methods. The bottom one is the new filtering we proposed. So as you can see, the one at the bottom doesn't have any spikes at the end of the figure, which means like it is producing less false positives, and it is quite uniformly distributed compared to the others. So as we can see, none of the normalization methods are making any difference, but the filtering seemed to make some impact on the accuracy of the results and the power of the test. But it has to be validated, and we are working on it with the simulated data. So what does Web Analyser provides us with in terms of differential expression analysis? You can fit a model by using eBias, which is the Lima-Wun pipeline, or you can use one of the HR pipelines like generalized linear model or generalized linear model with Cas-based, and you will have a table with all possible pairwise comparisons and differentially expressed up and down gene lists. You can download those as well. You can download everything you see in the application. And you can see an MD plot. You can produce a heat map. And you can produce also a Venn diagram, giving you the intersection between two different comparisons. And lastly, you can also run differential expression at gene level. To do that, we are using Fry to treat every gene as a gene set, because every gene has at least 6 guides, at least 4 guides. And using those guides in one set of gene, we do have applied Fry, and we can see the results for that. And RB1 gene is how we discovered that gene by using Fry. And it was a very meaningful result that caused us to publish that paper. So that's actually all, and conclusion and future direction. So there are 3 main points I wanted to make here. One of them is the new filtering approach can improve the downstream analysis, and we will be working on it. And bimodality of the count distribution is apparent in CRISPR screens and cannot be removed by filtering like it can be in RNA-seq. And this has to be taken into consideration when analyzing CRISPR screens. Maybe new methods has to be developed. And the impact of RNA-seq normalization methods from AGR is indifferentiable. When applied to CRISPR screen, a new normalization approach for the screens is needed as well, has to be developed. And my acknowledgements to Tony, Kate, who developed this R-Shiny tool, a Europe student, very dedicated and successful student. And Luke for his help and all the discussions about normalization, and we're still working on it. And Terry's insights. Marie is always helpful to discuss everything. And yeah, Marco Harold is the other head that I'm working with. They provide me all the data. Andrew gives us insights from a biological point of view. Yeah, and I thank to my consumer ready program. That's all. Thank you. If you have any questions, I'm happy to take now.
Thank you, Göknur. Questions? Just going up the back, Göknur, one moment.
No worries.
Apologies if this was answered in the talk, but it seems like your filtering method was really valuable. Do you have any plans to publish that separately so that it can be used like from R directly rather than through the Shiny?
Yes, we have the code. If you want it, I can send it to you at this moment, but we are writing a small manuscript on it to publish it as well. Yeah, I think it will be useful to apply in any data set with lots of sparsity in it, even the single cell. It might be applicable, but obviously with single cell, you have to define your conditions first, and then, I don't know, I haven't think about how to apply that there.
Thank you very much for your talk, Göknur.
Yeah, no worries. Thank you for everything.